So then the problem of how does it copy itself and how does it get used to make a protein that would cause green eyes. Now we're along those lines, right? So if we look at the first one, how does DNA copy itself? Watson and Crick took a stab at that with a short paper, but not much of the details. The details were worked out by several different people working at several different times. Here we have a key word that is a common word, but it's not commonly used, but it's the word that's used here with this idea. If I have some DNA, I'll make it double-stranded, right? And it's going to be used to make copies of itself. It's going to be used as a template. That's what that word means, like a pattern. It's going to be used as the pattern to make these copies. Okay, so describe the basic process. Here we go again. How basic do you want me to be? Because this can be very complicated or it can be basic. And the EOC test, they're only going to ask you basics. I will up the ante, no extra charge, and ask you some more details, right? How that relates to transmission and conservation of the genetic information. What do those words mean? Well, how the copying process, which we call replication, if we hadn't said that word. This word you need to keep straight from a couple other words coming later, right? The word that was chosen for DNA copying is replication. That's what I've shown right here, right? How does it relate to the transmission of genetic information? Well, if I have copies, I can send my kids some copies and I can still have a copy, right? So that's how it relates to that. And the conservation, what does that mean? The information in this DNA molecule is the same as that one. Remember, we said that's due to base pairing. The conservation of this exact information is due to base pairing. So here was Watson and Crick's basic idea, and here's the basic idea, and I think this is all the basic idea that you're going to get on the EOC test. But again, I'm going to educate you more than that. So here in dark blue, we have the molecule that they're calling the parent molecule, so to speak, because it's going to give rise to these two little babies over here, right? If this parent molecule unzips, Watson and Crick said, which would not be hard, catalyzed by an enzyme like you'll see, it involves breaking hydrogen bonds, which aren't too strong, right? But I'm going to say this while we're at it. This molecule is very stable because even though it's held together by weak hydrogen bonds, it's held together by a whole bunch of weak hydrogen bonds. So it's like a zipper very much. Here it's unzipped, right? The little teeth that had to break separate from each other, like the teeth in a zipper, to get unzipped were hydrogen bonds. But like a zipper, if you have a whole bunch of them, that zipper is held together pretty strongly. Okay, anyway, help of an enzyme, it unzips, and that, in the words that are commonly used, exposes these bases. They are no longer paired with each other, hydrogen bonded with their complementary partner. So it exposes the bases, and now in this cell, where this is happening, which would be the nucleus, right? DNA always stays in the nucleus, or in a prokaryotic cell, just in the cell in general, right? Nucleotides, again with the help of enzymes, get assembled. The base part of the nucleotide is going to connect to the only base it can connect to, A to T and C to G. And Watson and Crick said, if this is how it works, then that's what you're going to get, two copies. If you read each of these from top to bottom, they're just like this one over here, AT, CG, TA, AT, CG, TA. Why is that? Because of base pairing. Base pairing ensures exact replication. Now, what we're going to get to in the end of this is any kind of copying process is not perfect. And so if mistakes are made in this copying process, then we call them mutations. And they are the heart and soul of that process called evolution. Why things change. Things would never change if the information to make things was always copied identically. There could be no change. So mutations coming up are important. But for right now, the fact that this copying usually happens exactly correctly is due to base pairing. So we're going to watch this in more detail, right? And we're going to use some videos to do that in class. So this is going to be one of them. So here is one aspect of this replication process, that in a DNA molecule, the unzipping that we showed in a very short section of it happens in several places, right? We call them replication bubbles, right? And in each bubble, there are two forks where they're unzipping. That's called a replication fork, okay? This is a very simple model that the unzipping simply continues in each bubble until one bubble merges with another bubble. All the while, new nucleotides are being attached on both sides of the bubble to their complementary bases of what we are going to call the template strand, right? I should have said that in the last diagram, but like that last diagram, the original strand here was dark blue, and that's called the template strand. And now we have the newly put together strand, which doesn't have a specific name, but you get the idea. So here is an actual micrograph. This is about as good as you can take this. This is a very small, thin molecule. This is an electron micrograph of a replication bubble. Those arrows were pointing to two replication forks, right? This is the part that hasn't been unzipped yet in between, and here's another replication bubble. And in each of our pieces of DNA, which in your cells there are 46 of, right? Each chromosome consists of one piece of DNA. Each of those pieces, when it's being copied, has thousands of replication bubbles. But what we're going to look at is what happens only in one, okay? And we'll watch it in video in class. Okay, so I came back to add this one in here. These are all the dirty details of replication, okay? So this is a replication bubble. Thousands of them in a eukaryotic chromosome like yours and mine's, all working this same way at the same time to eventually have this whole long string of DNA copied, right? The strand, that's the original, that's being used to make the complementary other side is called the template strand, right? Remember? This part, right? This is the other template strand. These are the originals. They're going to be part of the copies, right? This is that replication kind of thing that's called semi-conservative. These two are going to be part of the copies, okay? And for that reason, I'm going to make the copies in blue. So here's what happens. Everything else is going to be in blue too. I drew this a little too high, okay? But first of all, one of the enzymes you need to know the name of is commonly represented like right here at the replication fork, right? Where the bases are still hydrogen bonded to each other to make this like a double-stranded ladder, but this is helping to weaken those bonds and break them. This again is helicase. Okay, it is unwinding and unzipping the double helix, helicase. Okay, so now what happens is at each place where this unzipped, and that's right in the middle of the bubble, right, where it started to unzip, a DNA polymerase is going to jump on, and it's going to start to move down the strand, and as it moves, it catalyzes the making of a new strand, complementary to the template strand, attaching nucleotides with a base that's complementary to the one on the template strand, A to T and C to G. Okay, so now I'm going to start this one on the bottom. If it's moving in that direction, then after a little while, it'll be over here. Okay, so I'm going to put it over there now, and if I can remember how this little thing works to make something erase. This thing is a pain. My fault, sorry. It's hard to find this little button. This is one of these human error kind of things. It's driving me crazy right now. There's a button, and that didn't do it. This is nuts. Ha! Okay, so I was going to start over, but I'm not. Okay, pretend this is not here. I'm doing it that way. It's now here. Okay, so it has put together a complementary strand, which I'm going to represent by just that string, right? So this is one of the new copies. It's half of the old one and half of a new one. If this one on the bottom is going in this direction, this one on the top is going in the other direction. That's the anti-parallel aspect of the template strand, that one side is upside down compared to the other. So when this one gets over here, for instance, it's not over here anymore, right? It will have put together this strand. Okay, so far so good? Okay. So first of all, what direction are they going in? Well, the way we talk about direction in terms of this DNA molecule has to do with the way that we number the carbons in the sugar molecule that's part of the sugar phosphate backbone, right? And the way that it works is that this enzyme is always traveling along the template strand in the direction of the template strand's 3' toward its 5' end. Those numbers refer to the number of a carbon molecule in the ribose, that in class I'll show you all this. So since this template that I drew in red is anti-parallel, that means for this bottom strand, that's the 3'. That's called, that little apostrophe mark is called a prime mark. The 3' to 5' end. This DNA polymerase is moving from the 3' end of this bottom template strand toward the 5' end, and this one at the top is moving along the template strand from its 3' end towards its 5' end. That's the direction that causes this to be happening in opposite directions at the same time in every single replication bubble. Okay, so now what's happening over here behind it? Well, behind it, as this continues to unzip, we get new polymerases jump on. I'm going to put 2 of them here. They jump on and they start working along the template strand in the only direction that they can. And as time goes by, again, they're going to move. So this one right here, let's say it started over here. So it's already put together this little string. And this one is going to start to put together another little string. And eventually, these are all going to bump into each other. And this one, again, is eventually going to bump into where this first strand started getting made. And eventually, very simply, all these gaps will be filled in. These little pieces, while they're still little pieces, are called Okazaki fragments, named after the guy that figured them out. That this is how it works. This was in the 1970s. Okay? And this is also happening on the other side. So now I'm going to add this, the last thing we have to add. These strands that I first talked about, this one that got made in that direction right there, and this one that got made in that direction right there, the ones that are being made continuously, not in little fragments that are going to get added together, like these Okazaki fragments, but are being made continuously, they're called the leading strand. Leading strand. What are they leading? They're in front of leading this strand that's getting put together in pieces. That eventually all these pieces will be connected together, but this strand that's being put together in pieces is called, because it's behind, is called the lagging strand. Lagging means you are behind. Oops, lagging strand. So now over here, we have the same thing happening as down here, except in the opposite direction. We have RNA polymerases that have jumped on. Let's put one, let's say it's gotten here. It's putting together that Okazaki fragment. Here's one that jumped on over near the fork, and it's put together this Okazaki fragment. So this is a leading strand on the top end. And this is collectively the lagging strand being put together on the bottom end, being put together in pieces. Pieces we call Okazaki fragments. This takes a while to digest in your head, but it's very mechanical and very repetitive. So if you replay this slide a few times, it should all sink in with these terms, okay? So this is definitely above EOC stuff, but it's not above honors biology level stuff. So that's why you're going to see a little bit of it on your test. This directionality that's involved with the copying replication of a DNA molecule because of its anti-parallel structure. That's why these strands are being replicated in opposite directions, and that's why this system had evolved to where you have a continuously formed leading strand in front of a formed in segments lagging strand. Make sense? I hope.